a very good day to Dr. Haslina. So this is the presentations for Bioinformatics 1 Gene Projects. And this group is consists of me, WELSON, WOONG, and my group members, KWAN. So this is the table of contents where it will be divided into three, where there's a gene overview, methodologies, and result, and finally the discussions. So this is the gene overview. So the gene that we select for this project is CYP10, also known as the sialic acid binding, which is a Ig like lectin 10. It is found in the Homo sapiens with the reference sequence number NP149121. So it functions as an inhibitor's immune receptor that regulates immune response via sialic acid and dependence binding, especially on innate immune cells like B cells and macrophages. So this is the methodologies that we use and the tools throughout this project. So we use T+N from NCBI and BLASTP from NCBI, also MAFFT for the pair sequence alignments, and then using Mega for phylogenetic tree. So we will go through T+N first. So firstly, we retrieve the human siglet 10 protein sequence from the NCBI database using the reference sequence of ID NP149121.2. So then we use T+N to compare this sequence against the EST database of Staphylococcus trophicalis to find potential mRNA transcript that align with human siglet 10. So this is the result of the descriptions and the graphical summaries for the result of T+N. And then this is the pairwise alignment of the T+N's result. So next, we're moving on to what is novelty criteria. So to determine whether the gene is truly novel or not, we use the following criteria, which are if a match is 100% identical to a non-gene in the same species is not novel. Hence, it must be less than 100% or matches a different species, then it will be considered a novel. So using this definition, we will evaluate our novelty of our candidate sequence. So the novel proteins. The candidate sequence came from the Staphylococcus trophicalis and its taxonomy classified it deep within the amphibians lineage. This emphasized its evolutionary distance from humans and strengthened the potentials of novelty of the gene. So I will talk about BLASTP. So using BLASTP, we will be compare the protein translated from our candidate sequence against a non-redundant protein database. So this step will ensure that our sequence is already characterized in Staphylococcus tropicalis. Hence, we can see that the BLASTP result. So the BLASTP result shows no 100% identity matches in Staphylococcus tropicalis. So this confirms that our sequence is novel and is likely representing an annotated siglet 10 like gene in amphibians. So the novelty is 87.76%. So that's all for my part, and I will pass to my partner, KWAN.
Hello and good day everyone. And for my part, I will continue with the explanation for the multiple sequence alignment part and the phylogenetics section for this project. So what is MAFT? MAFT stands for multiple alignment using fast Fourier transform. It is an iterative approach for multiple sequence alignment and is widely considered as one of the most accurate and versatile tools for multiple sequence alignment. For this math section, we align the and we align the sequences from the novel protein that we have obtained, which is the Xenopus protein, and then compare it with the other Siglec-10 sequences, which we obtained from human, rat, koala, gorilla, and II. The alignment showed that the Xenopus protein matches mostly to the VZ immunoglobulin-like domain, suggesting it might be a partial sequence or a unique isoform. You can see the sequence here. Crystal format alignment using the math tool in the Amble EBI. Next, we move on to the phylogenetics section of the project. In bioinformatics, phylogenetics is a study on the evolutionary relationships of genes and proteins. We can thus infer the relationship of genes or proteins by either building a parsimony tree or a distance-based tree. For this project, we use the one of the distance-based tree, which is the neighbor joining tree. Here is what the neighbor joining tree style looks like for the results that we have acquired through math. This tree is built using the Mega 12 software, which we can use the math to align it to carry out the multiple sequence alignment before building this tree. This is the bootstrap and J tree style, the topology only tree style, and the bootstrap consensus tree style. Now for the discussion part. In summary, the novel sequence from Xenopus tropicalis shows around 30% identity with human Siglec-10, meeting the novelty threshold, and also appearing and also that is not far off from the definition of homology, which means that Xenopus tropicalis sequence here is a type of homologous sequence with the Siglec-10 found in humans. The MS multiple sequence alignment revealed the conserved domains, and the phylogenetic tree positioned it as an early diverging homologue of the Siglec-10 sequence. This gene could play an immune regulatory role in amphibians, like its mammalian counterparts. Our analysis shows how bioinformatics can help identify uncharacterized genes in understudied genes in other organisms within the family. And that is all from me. Thank you.